So good day everyone. We are the group Orange from BSCHEM 31 and we will be discussing the lipids major metabolic pathways part 2. The lipogenesis and its regulation will be discussed by Mary Rose Arce and yours truly, John Mark Leonid. Esterification and lipolysis to be tackled by Zenry Mico Terrado and Rain Grace Reyes. And finally, the regulation of fat metabolism will be discussed by Gabrieli Monique Albornoz and Peaches Aliana Martinez. So to give you an introduction to lipogenesis, we have Miss Mary Rose Arce.
So thank you. Lipogenesis is a process at which carbon precursors of acetyl-CoA are synthesized into fatty acids. So what are those precursors? This includes the glucose. Thus, the lipogenesis occurs during the fed state, since we know that during the well-fed state, that there is a high amount of glucose being absorbed. And by the time when there's excess amount of glucose in our blood, our body will produce insulin, which will regulate the levels of the glucose. Also, the hormone insulin triggers our body cells to consume or use up the glucose for metabolic processes. When high amounts of glucose are being absorbed in the body, consequently, high amounts of ATP are also being produced. The lipogenesis is an anabolic process. It uses up ATP to produce fatty acids. With this, our body also has its own way to regulate the production of ATP to prevent the overproduction of that energy currencies. And how does this happen? The acetyl-CoA, which is derived from pyruvate and from glucose, will be synthesized into fatty acids to store energy for future use. Now, the lipogenesis is present in the tissues such as liver, kidney, brain, lungs, mammary gland, and adipose tissue. Inside the cell, its process takes place in the cytosol. Remember that acetyl-CoA is the starting precursor or the immediate substrate, while the palmitate is our product here in lipogenesis. And for the cofactor requirements, it includes the NADPH, ATP, manganese 2, biotin, and bicarbonate, which will be our source for carbon dioxide. So to give you the complete metabolic pathway of lipogenesis, let's give the floor to John Mark Bilioni.
So thank you, Mary Rose. Here, what you can see in this figure is the simplified overview of lipogenesis. And as said earlier, acetyl-CoA is our initial precursor here. And where do we get acetyl-CoA? It's from the inside of your mitochondrion. However, acetyl-CoA cannot be transported out of your mitochondrion directly. Instead, the citrate, which is one of the intermediates of your citric acid cycle, can be transported out via the tricarboxylate transporter. Now, in the cytosol, citrate will be degraded into oxaloacetate and acetyl-CoA through the help of the enzyme ATP citrate lyase. Now, acetyl-CoA will be carboxylated to malonyl-CoA with the help of acetyl-CoA carboxylase or ACC enzyme. And this is what we want to emphasize. This is the committed step, and therefore it is the one that is rate determining and highly regulated. Now, acetyl-CoA and malonyl-CoA, with the help of enzyme fatty acid synthase, they will form fatty acid chains until the acyl chain is now corresponding to palmitate, which is our end product here. Bear in mind that the fatty acid synthesis or lipogenesis takes place for 7 cycles until it forms the palmitate product. Now, the lipogenesis also needs reducing power, which is the NADPH. Now, this is the comprehensive overview of our lipogenesis. So let me zoom into you. So as you can see here, our initial precursors are acetyl-CoA and malonyl-CoA. And through steps in lipogenesis, the fatty acid chain will form. And then the process of lipogenesis will happen for 7 cycles until this acyl chain is now containing 16 carbon units corresponding to our product palmitate. Now, before discussing the detailed steps or detailed mechanism of the lipogenesis, let's discuss first how malonyl-CoA is formed in the committed step. So here, as said earlier, we need bicarbonate as the source of carbon dioxide. And in the presence of ATP, the coenzyme biotin becomes active, meaning carboxylated. Now, with the action of your acetyl-CoA carboxylase, which can be activated or inhibited through biosubstances, which you will see in the later discussion, the acetyl-CoA will be carboxylated to form your malonyl-CoA. So in short, the coenzyme biotin here acted as the carboxyl group donor. Now, going back to the discussion of the detailed mechanism or comprehensive overview of lipogenesis. So here we have acetyl-CoA and malonyl-CoA as our starting product here, and they will attach to the multi-enzyme complex of fatty acid synthase. So enzyme 1, let's assign it as cysteine, while enzyme 2, let's assign it as pantotene. Now acetyl-CoA will attach to enzyme 1 while malonyl-CoA to enzyme 2. And in this process, 2 CoASH are given off. So as you can see here, in the step 1, we have 5 carbon units attached to the enzyme 1. So 2 from acetyl-CoA and 3 from malonyl-CoA. However, in the step 2, through the help of trichetoacyl synthase, this acetyl group will transfer to your malonyl chain, forming 4 carbon units. How is this so? Since in the step 2, carbon dioxide is being released. So 5 minus 1, that's only 4. So all in all, your malonyl-CoA contributes only 2 carbon units in each cycle. Now in step 3, the trichetoacyl reductase will reduce this product into this one. So here you can see that from carbonyl group, it becomes hydroxyl. Since it's a reduction, it needs reducing power, which is the NADPH. Now in the step 4, with the action of your hydratase, it will form the unsaturated acyl enzyme. So as you can see here, we have a double bond. And then in the step 5, the enol reductase will form the saturated chain of acyl enzyme, and this one is now containing 4 carbon units. And since it's a saturation process, therefore a reduction, it needs another reducing power, NADPH. Now the question is, after forming this 4 carbon unit acyl chain, what will happen to this? It will transfer back again to your enzyme 1, to the enzyme 1, leading to the second cycle of your lipogenesis. So instead of having acetyl group here, we now have a 4 carbon acyl chain. And then another unit of malonyl-CoA will attach. And the process of lipogenesis will repeat for 7 cycles until this acyl chain is corresponding to palmitate, meaning containing 16 carbon units. And what will happen is that after forming the palmitate chain, the thioesterase will liberate the free palmitate, forming again our fatty acid synthase multi-enzyme complex with no attached substrate in it. So, for summary, here it is. As I said earlier, there are 7 cycles involved in the lipogenesis. So starting from acetyl-CoA and malonyl-CoA, it will form the 4 carbon unit chain acyl chain. So in this process, we needed 2 NADPH and 2 H plus, and it generated CO2, 2 CoASH, 2 NADP plus, and H2O. And after forming the acyl chain, we needed again 1 unit of malonyl-CoA, resulting to 6 carbon unit acyl chain. So in this step, we needed 2 NADPH and 2 H plus, CO2, and 1 CoASH only, since malonyl-CoA is the only precursor here in cycle 2 containing CoA. And then 2 NADP plus and H2O are released. So as you can see here, after each cycle, 2 carbon units are being added to the acyl chain. So similar steps will happen to the subsequent cycles. So all in all, the summary of the reaction would be 1 acetyl-CoA, 7 malonyl-CoA, 14 NADPH, 14 H plus yielding 1 palmitate, 7 CO2, 8 CoASH since 2 from cycle 1 and 1 each from cycles 2 to 7. Therefore, 2 plus 6, that's 8. And then 14 NADP plus and 7 H2O. Now, to discuss to you how the lipogenesis is regulated, let's give the floor to Mary Rose Arce.
So for the regulation of lipogenesis, we will specifically discuss the regulation of the enzyme used from our committed step, which is the acetyl-CoA carboxylase. The acetyl-CoA carboxylase is an allosteric enzyme, and its known activator is the citrate. So the citrate activates your ACC complex from the inactive dimer to form the ACC polymeric form. With this, the acetyl-CoA carboxylase in polymeric form can now function. Aside from citrate, your acetyl-CoA carboxylase is also activated through the hormone insulin. How? The insulin favors the phosphorylation of AMPK by making it inactive. Take note that when there is a low amount of AMPK active, the phosphorylation of acetyl-CoA carboxylase is favored through the help of the enzyme protein phosphatase. And if acetyl-CoA carboxylase is dephosphorylated, it is now active, then ACC enzyme can now process the conversion of acetyl-CoA to malonyl-CoA. Through subsequent process of fatty acid synthesis and elongation, the acyl-CoA will be formed. And now, if there is a high amount of acyl-CoA being synthesized without esterification, though don't worry, since our body has its own way to inhibit the lipogenesis. But again, how? We need to increase the amount of AMPK again by making it active. So we can increase it by first, your acyl-CoA together with the hormones glucagon and epinephrine activates your CAMP. And then your CAMP activates your CAMP-dependent protein kinase, which in turn activates your AMPKK. So what does this AMPKK do? It phosphorylates your AMPK, making it active. Then the activated AMPK will phosphorylate the acetyl-CoA carboxylase, and thus it makes it inactive. So that's the inhibition process where we can regulate the excess acyl-CoA being produced. So in summary, the stimulator of lipogenesis are insulin and citrate. And this happens during the fed state, while the inhibitors are the hormones glucagon, epinephrine, together with the acyl-CoA. This happens during the fasting when our body already needs the stored energy. So the question is, you may be asking, what is happening to the acyl-CoA or the free fatty acids being regenerated from lipogenesis? Actually, it is further esterified to form triacylglycerides to store energy by esterification. And when our body needs the energy from the stored TAGs, lipolysis will now take place. The process of esterification and lipolysis will be discussed by the next set of reporters, Zenremy Coterado and Rain Grace Reyes.
Good day, I'm Grace Reyes, and I'm going to report about esterification in lipids and the major metabolic pathways. So first of all, what is esterification? Esterification, also known as pitcher esterification, is the esterification of a carboxylic acid by heating it with an alcohol in the presence of a strong acid as the catalyst. So as you can see here below, we have the mechanism for esterification. We have here a carboxylic acid and an alcohol, which results in the formation of ester by the presence of strong acid as the catalyst. Here we have the sulfuric acid as an example of a strong acid. So in lipids and its major metabolic pathways, we can see the esterification process through the synthesis of triglycerides. So what exactly is a triglyceride? Triglycerides are a type of fat or lipid found in your blood. When you eat, your body converts any calories it doesn't need to use right away into triglycerides. The triglycerides are stored in your fat cells. Later, hormones release triglycerides for energy between meals. It is also called triacylglyceride or triacylglycerol, a major storage form of lipid in the body, composed of a glycerol backbone and 3 esterified fatty acids. As you can see here, we have the glycerol and 3 fatty acids, which results in the formation of triglycerides. So the major building block for the synthesis of triglycerides in tissues other than adipose tissue is glycerol. So adipocytes lack glycerol kinase, therefore dihydroxyacetone phosphate or DHAP, which is produced from glucose during glycolysis here, with the glucose during glycolysis, is the precursor for triglyceride synthesis in adipose tissue. The vast majority of the glycerol-free phosphate is derived from the glycolytic intermediate dihydroxyacetone phosphate here by the action of cytosolic NAD-linked glycerol-free phosphate dehydrogenase here. In the liver and kidney, a small amount of glycerol-free phosphate is also formed from glycerol by the action of glycerol kinase here. So these two processes both result in the formation of L-glycerol-3 phosphate. The other precursors of triacylglycerols are fatty acyl-CoAs, formed from fatty acids by acyl-CoA synthetases, the same enzymes responsible for the activation of fatty acids for beta oxidation. The first step in the biosynthesis of triacylglycerides is the acylation of the two free hydroxyl groups of L-glycerol-3 phosphate by 2 molecules of fatty acyl-CoA to yield the acylglycerol-3 phosphate, which is more commonly known as phosphatidic acid or phosphatidate. So here we have the L-glycerol-3 phosphate, and we have here the two fatty acyl-CoAs. Here we have one fatty acyl-CoA and one fatty acyl-CoA here. All can be converted. So these two will link with the L-glycerol-3 phosphate with the help of acyltransferase to create phosphatidic acid here, or diacylglycerides, because we have two free fatty acids, fatty as acyl-CoAs. And so phosphatidic acid is found in small amount in cells, but is a central intermediate in lipid biosynthesis. It can be converted either to a triacylglyceride or to a glycerophospholipid. So here in the second step of triacylglyceride biosynthesis, phosphatidic acid thus formed is hydrolyzed by phosphatidic acid phosphatase. Here it hydrolyzes a phosphatidic acid to form a 1,2-deacylglycerol. Here, 1,2-deacylglycerol. So here on the other hand, we have the phosphatidic acid, which is converted into glycerophospholipid. So the next step is, okay, so the acylglycerols obtained from step 2 here, the acylglycerol here, are then converted to triacylglycerols with the incorporation of a third fatty acyl-CoA by an enzyme acyltransferase here. Another one, so it's just the same process, but this time we add the third fatty acyl-CoA, which is why it's now called a triacylglycerol. So this process is called trans-esterification. So if we all remember here, let's go back, we have the L-glycerol-3 phosphate. So it is not shown here, but here we have the phosphate group. So yeah, we have the phosphate group there. Okay, so here the phosphate group here, to remove that, we have to use the enzyme called phosphatase. So the phosphatase basically breaks the bond between the oxygen from the glycerol and the phosphorus from the phosphate group. So we could easily attach the fatty acyl CoA. So in short, the linkage of the three fatty acyl CoAs with glycerol is the process of creating a triglyceride. So these are my references, and thank you for listening. The next part will be reported by my groupmate.
Hello, good day everyone. I am Zenry Mico Terrado, and for this presentation, I will be discussing one of the topics in Biochemistry 2, which is the lipolysis pathway. For the general definition, lipolysis is a metabolic process in which triglycerides are hydrolyzed into glycerol and free fatty acid. Lipolysis utilizes stored energy during fasting or exercise and typically takes place within our adipose tissue. So take note of the terms mentioned in the definition because it is necessary to familiarize ourselves within the terms in order to fully understand the concept of lipolysis. And these terms include the chemicals and the processes such as the triglycerides, hydrolyze, the glycerol, fatty acid, adipose tissue, and so on. We must have at least an idea of what these terms mean by now. To provide an overview of the content of the discussion, we will first go deeper in defining lipolysis and learn about the chemicals involved in the process. Second, I'm going to discuss the process of breaking down triglycerides step by step and also the enzymes that drive the process. Next will be the endocrine control of lipolysis. This involves the hormones that triggers the mechanism. And last will be the lipolysis pathway in adipose tissue. So to further understand the term lipolysis, let's split the word. So lipo meaning lipid, which is nothing but fat, then lysis means breakdown. So basically, lipolysis means breaking of fat to produce energy. We also have this misconception about fat because maybe many of us have seen articles or videos on the internet claiming that you can burn fat. However, this is just a myth or a misconception nga, since fat cannot be burned, hence it can only be utilized. Again, you cannot burn fat, nor can anyone else, but you can utilize or produce fat. So that is the proper approach or correct way of addressing fat loss. Now let's define some important chemical compounds and process in lipolysis. So first is the triglyceride, or TG. It is the main constituent of vegetable and animal fats in the diet and are the main constituent of the body fat stores. So TG stores in adipocytes undergo continuous lipolysis and esterification, with the two processes utilizing separate pathways, reactants, and enzymes. So in other words, yung lipolysis and esterifications are inverse processes because yung lipolysis is the process by which fats are broken down, while naman sa esterification, it's the process by which fats, specifically yung mga triglycerides, are formed. So next is hydrolysis. So take note or remember also of this process since this is important in the mechanism of lipolysis. So hydrolysis, we can define it as the reaction of an organic chemical with water to form two or more new substances. It is one of the responsible for breaking down of TG or the triglycerides. So upon doing lipolysis, free fatty acids and glycerol are the end product of the mechanism. So triglycerides have the structure of a glycerol bound to three fatty acid molecules. So we all know naman na yung alcohol, meron siyang hydroxyl groups, whereas yung organic acids ay nagko-contain ng carboxyl group. So as a result, if mako-combine natin yung alcohol and organic acid, it can form an ester. And take note that triglycerides are also known as triesters. So for the definition, glycerol, this is the type of alcohol that can be easily converted into glucose, making it a good source of energy. As you can see here in the picture, here is the glycerol. It serves as the backbone of the triglyceride. Then next is the fatty acid tails. These are the organic acids naman, and each one of the three fatty acid molecules undergoes an esterification with one of the hydroxyl groups of the glycerol molecule. So here, the picture you can see, here are the fatty acid tails, 1, 2, and 3 fatty acids. And the last part is the ester bonds. These three ester bonds link the glycerol backbone to the free fatty acid tails. So ester bonds are formed as the result of the condensation reaction between glycerol and the fatty acid. So here the picture is the ester bond. It's the link between the glycerol and the fatty acid, 1, 2, 3. Now let's move on to the process of triglyceride breakdown. This can be done by breaking down TG to release the fatty acids from the monoacyl glycerol while simultaneously secreting lipases and bile rather. So we all know na lipases are enzymes that can break down fats in food, whereas naman yung bile is a digestive fluid that it can also break down fats into fatty acids. In order for breaking down of triglycerides to occur, each of the ester bonds needs to be cleaved by the process termed hydrolysis. So the whole process requires 3 separate steps. Each step or process cleaves one fatty acid away from the glycerol backbone until all fatty acids are liberated. So as you can see here in the first picture, it is the structure of the triglyceride. So the glycerol backbone is written in white, while the ester bonds are the pink one. Then the fatty acids are colored in yellow. So the first step is to hydrolyze the first ester bond here. So as shown in the second picture, the blue hydrogen atom on the far left, this one, bonds onto the oxygen of the glycerol backbone, while the remaining OH bonds to the carbon on the fatty acid residue on the right. So this second image demonstrates how the first step liberates the first fatty acid from its parent glycerol backbone. So in other words, itong original triglyceride has lost one of its fatty acid to become a diglyceride. So meaning, yung first product ng ating first step is diglyceride. Now look at the third picture, this one. It shows the second step for breaking down diglyceride. Again, this involves the hydrolysis, but for now, for another ester bond. In this case, it is the terminal bond on the other side. So kung kanina dito tayo sa first ester bond, we're now focusing on the terminal bond, and then yung opposite side, and this one. And this step liberates further fatty acid away from its parent glycerol backbone, or to put it in another way, the original triglyceride has lost one of its fatty acid to become a monoglyceride. So the second product for the second step is monoglyceride. Then for the third and the final step, here this picture, again, hydrolysis ulit for another ester bond, but in this case, it is the middle ester bond. Through hydrolysis, it liberates a third and the final fatty acid away from its parent glycerol backbone. And the original monoglyceride lost its one fatty acid to become the alcohol known as the glycerol. So now we have the end product glycerol, and the 3 fatty acids are liberated na, and we can call it an FFA or the free fatty acid. So the end product for the last step will be glycerol and free fatty acid. So each step in breaking down triglyceride requires an enzyme. So for the first step, adipose triglyceride lipase or ATGL is required to catalyze hydrolysis of triacyl glycerol, TAG, also known as the triglyceride, to diacyl glycerol, DAG, or also known as the diglyceride. Next is the second step. It requires hormone-sensitive lipase, HSL. It is required to catalyze hydrolysis of diglyceride to monoacyl glycerol or MAG. It's also known as monoglyceride. Then last step, it requires monoacyl glycerol lipase or MGL. It is to catalyze the hydrolysis of monoacyl glycerol MAG to glycerol alcohol. Now let's move on to the third part of the discussion, which is the endocrine control of lipolysis. So endocrine system takes part in lipolysis since the rate of free fatty acids released from adipocytes is affected by several hormones. And we all know the hormones are chemical messengers in our body. They circulate rather through our bloodstream to tissues or organs. So some of the hormones are classified as lipolysis activators. That is, they are responsible for initiating or triggering the said process. So the most potent hormones in lipolysis appear to be the catecholamines. These are the epinephrine or norepinephrine, or commonly known as the adrenaline. We also have the thyroid hormones, specifically the T3 and T4, and the glucocorticoids. Example is cortisol. So there are other circulating mediators or hormones reported to augment the effect of the primary stimuli. It includes the growth hormone, glucagon, thyroid stimulating hormone, alpha and beta melanocyte stimulating hormone, adrenocorticotropic hormone, and so on and so forth. So when thyroid hormones itself are passed through the adipocytes, there will be no free fatty acids will be released, as shown here in the illustration. This is the thyroxine that then passed through the adipocytes. There will be no FFA release. On the other hand, when catecholamines are passed through the adipocytes, small amount of free fatty acids will be released, as also shown here in the illustration. So catecholamines act rapidly in promoting lipolysis by stimulating membrane-bound adenyl cyclase, thus increasing intracellular levels of cyclic AMP, or also known as CAMP. So through increasing the number of beta-1 adrenergic receptors on adipocytes, thyroid hormones become synergistic with the catecholamines in promoting lipolysis. So synergistic refers to be the interaction or cooperation of two or more substances to produce a combined effect greater than the sum of their separate effects. So for the case here, there is a synergistic effect in thyroxine and epinephrine plus the cortisol to have a large amount of free fatty acid released. Now let's move forward to the fourth part and the last part of our discussion, the lipolysis pathway in adipocyte or adipose tissue. So this pathway refers to the cellular mechanism for lipid mobilization from fat stores. So we have here the diagram for the pathway of lipolysis in adipose tissue. So we are going to start here in triglyceride, which undergoes lipolysis by the process of hydrolysis. So it will give us an end product of glycerol and free fatty acids. So these two will be mobilized from adipocyte into our blood. But the release of free fatty acids from adipocyte will be dependent upon the resultant of the process, which is nakaka-apekto dito is yung mga several hormonal influences. So the FFA will be associated with the enzyme adenyl cyclase. So here, we'll travel from FFA from here to the adenyl cyclase. We also have here the potent hormones for lipolytic activity. So through increasing the number of beta-1 adrenergic receptors, the synergistic effect of catecholamine plus the thyroid hormones and the other hormones will be a good help in stimulating the adenyl cyclase. So here we have this catecholamines, the thyroid hormones, and the other hormones that can stimulate the activity of the adenyl cyclase. On the other hand, meron ding mga nako-consider na anti-lipolytic agents such as the prostaglandin E2 or the PGE2 and the ketone bodies. Nire-reduce nito yung activity ng adenyl cyclase. So, however, these lipolytic agents and anti-lipolytic agents na na-mention are both nakakatulong for the generation of cyclic adenosine monophosphate or CAMP. So again, the enzyme adenyl cyclase with the several agents convert adenosine triphosphate or ATP to its derivative cyclic adenosine monophosphate or CAMP. So from the CAMP, by this intracellular second messenger, it will stimulate a CAMP-dependent protein kinase. Best example is the protein kinase A. So pinapataas nito yung cytoplasmic levels ng CAMP, and this is responsible for converting inactive HSL or the hormone-sensitive lipase into its active form. So as you can see here, the ATP is converted to ADP in order to the inactive HSL be converted to its active form, HSL. And yung active form ng hormone-sensitive lipase is the phosphorylated form, and thus it promotes again lipolysis. So this will go back as to the first process. So here, the phosphorylated form, it will promote again the process of lipolysis, and we will go back to the first process. So for summarizing the whole pathway, first, the TG stores of fat tissue are continually undergoing lipolysis in the process of hydrolysis and reesterification. So these two processes, for the most part, utilize separate reactants and enzymes, thus allowing several metabolic endocrine factors that regulate the metabolism of this tissue type to act upon one process or the other. So yung resulta ng dalawang process na ito ay nagdedetermine ng amount of free fatty acids na kayang ma-release, which can exert profound effects upon the metabolism of other tissues, most notably sa kidneys, sa livers, and other muscle tissues. Next, the initial event in the mobilization of free fatty acid from adipocyte is the hydrolysis of TGs by the primary enzymes, an event referred to triglyceride breakdown. So again, these primary enzymes include the adipose triglyceride lipase or the ATGL. We also have the hormone-sensitive lipase or HSL, and lastly, the monoacyl glycerol lipase or the MGL. So next, the HSL of fat tissue is activated by epinephrine and cortisol, and the thyroid hormones, specifically the T4 and T3, are synergistic with epinephrine. So synergistic in the sense that they stimulate the synthesis of beta-1 adrenergic receptors on the plasma membranes of adipocyte. Next, the epinephrine acts through the cyclic AMP, which in turn activates an intracellular CAMP-dependent protein kinase. So this kinase aids in the phosphorylation of HSL, which is required, again, for its activation. Di ba kanina, from the inactive HSL, it will be converted into active one through the help of phosphorylation. Next, the glucocorticoids promote lipolysis through a CAMP-independent pathway. So it is by promoting gene transcription and by following the intracellular receptor stimulation, yung HSL, the HSL biosynthesis will be increased. And lastly, the insulin reduces the rate of free fatty acid release from adipocyte. So ito naman, it can happen in different mechanisms. It can be stimulates the activity of CAMP-dependent phosphodiesterase, which nare-reduce nito yung intracellular CAMP concentration. Pwede rin naman na yung insulin, it activates the specific phosphatase, which dephosphorylates. Di ba nga, yun nga kailangan ng phosphorylation para ma-active yung HSL. But yung insulins, nakaka-affect sila to dephosphorylate HSL, so thus returning to its inactive form. And lastly, maybe it antagonizes the actions of the glucocorticoids on the adipocytes. And that will be all. Thank you.
The last topic to be discussed is the regulation of fatty acids beta oxidation, to be presented by Picha Saliano Martinez and Gabrieli Monique Albornoz. To introduce the fatty acid oxidation, it mainly occurs in the mitochondria and involves a repeated sequence of reactions that result in the conversion of fatty acids to acetyl-CoA. All of the enzymes involved for oxidation is found in the matrix of mitochondria. Also, we have to take note that it yields 4 ATP per round of oxidation. It is in the form of FADH2 molecule, NADH molecule, and acetyl-CoA molecule. Mitochondrial beta oxidation of fatty acids requires 4 steps, and all of which occur in the mitochondrial matrix to produce 3 energy storage molecules that was mentioned earlier per round of oxidation. First is the acyl-CoA dehydrogenase. The second step is the enoyl-CoA hydratase. Third is the 3-hydroxyacyl-CoA dehydrogenase. Lastly, the 3-ketoacyl-CoA thiolases. Let us now see how the oxidation occurs.
So the fatty acid is activated to fatty acyl-CoA by acetyl-CoA synthetase as catalyst. Then the acyl-CoA will be substituted to carnitine, forming fatty acyl-carnitine by carnitine-acyl transferase 1. After that, acyl-carnitine will then move into the matrix by acyl-CoA and is catalyzed by carnitine-acyl transferase 2. Then, the free carnitine can move to the intermembrane space to the outer membrane space, allowing to repeat the process. So the fatty acyl-CoA will undergo beta oxidation. Then the rate-limiting step is the reaction involving carnitine-acyl transferase 1. Malonyl-CoA inhibits this step by signaling that there is excess energy after the concentration. So the excess energy will then be converted to fatty acids for storage. There are 2 enzymes of beta oxidation that is also regulated. First enzyme is beta-hydroxyacyl-CoA dehydrogenase. If the concentration of NADH is high, it's going to inhibit beta-hydroxyacyl-CoA dehydrogenase, since high amounts of NADH signals that the cells has enough energy, and the other enzyme is thiolase. So acetyl-CoA is going to inhibit thiolase. Now that we understand what inhibits it, let us now discuss what activates the process. So AMP is going to activate AMP-activated protein kinase, and this is going to phosphorylate acetyl-CoA carboxylase, inhibiting malonyl-CoA formulation. So when the concentration of malonyl-CoA decreases, there would be no inhibitor for the carnitine-acyl transferase 1, activating the beta oxidation. So again, as the concentration of AMP increases, breaking down ATP, it will cause for the activation of beta oxidation process.
The pathway that was discussed by Ms. Albornoz, let us now integrate it with the fatty acid synthesis. Malonyl-CoA inhibits carnitine-acyl transferase 1, and the concentration of NADH inhibits the enzyme in beta oxidation, the beta-hydroxyacyl-CoA dehydrogenase. Lastly, acetyl-CoA also inhibits the beta oxidation enzyme thiolase. Again, we have discussed the regulation of fatty acids beta oxidation. That is all for the presentation of our group oral report. Thank you for listening.